Good evening, Sandberg students, and welcome to Chapter 3. So tonight we're going to be talking about cell structures and their functions. We'll look at bacterial cells, plant cells, and animal cells. Three of the most common cell types. To discuss the cells, we first want to bring up something we've talked about in the past, cell theory, which states that all organisms are composed of cells. So living things are composed of cells, and only cells can produce new cells. The individuals that get the credit for this, Robert Hooke, who introduced us to cells when he was looking at cork and came up with that term. And then Matthias Schleiden, Theodor Schwann, and Rudolf Virchow are the ones who actually composed the cell theory. This is much later than Hooke's time. So the cell is the basic unit of life, and cells are what function in organisms. We are composed of millions of cells, and each of our cells has specific functions. If those cells aren't functioning properly, then our body won't function properly. Our organs won't function properly. Cells always have a plasma membrane. All cells have a plasma membrane that surrounds them. The structures within the plasma membrane are very similar in all cells, but may vary to some degree. All cells have protein molecules inside of them, inside of the plasma membrane. This is called an integral protein that spans the entire plasma membrane. This is a peripheral protein that's only found on one side or the other side of the plasma membrane. Each protein will have different functions. These are phospholipids. These are the most common structures found in plasma membranes. They're composed of hydrophilic, water-loving glycerol heads, and hydrophobic, water-hating fatty acid tails. So the water-loving heads interact with either the extracellular or intracellular environment, and the tails stick together. I just said this. The structure of the plasma membrane gives the plasma membrane its ability to be selectively permeable. When I think of the plasma membrane and selective permeability, I always like to think of a bouncer at a bar that can allow certain things through but keeps other things out. So it's like, you can go, but nope, you can't go. You can go, yep, come this way, but nope, you're not allowed to go. So the things that are allowed to go through the membrane are going to be small and nonpolar molecules, while anything that is too large to pass across the membrane or anything that is water-loving is not going to be able to go directly across the membrane. The plasma membrane contains, this is weird, sorry. Okay, the plasma membrane also contains proteins. Proteins can be used as receptors or for binding or for transport, or they can act as enzymes, which we'll talk about a little later, I think chapter 6. There's going to be, depending on the type of plasma membrane, you're going to have some form of a sterol that will stabilize and strengthen the membrane. For animal plasma membranes, we have cholesterol. But in bacterial plasma membranes, they have a different molecule. It's called a hopanoid, H-O-P-O-N-O-I-D. And in fungus, we have ergostrol. So there's a different form of the sterol molecule. that stabilizes the membrane, but they all have a stabilizing molecule. This is animal plasma membrane. So here we have the phospholipids. Some of the phospholipids have these little carbohydrate groups attached to them. That would be called a glycolipid. If the carbohydrate is attached to a protein, it's called a glycoprotein. These little purple structures here and here, those are the cholesterol molecules that strengthen and stabilize the membrane. This is the phospholipid bilayer. You have the outside and the inside of the membrane. And then you have the cytoskeletal filaments that are going to attach to the plasma membrane. And they help to give the cell more structure. So I said that the molecules that move across the membrane are small or uncharged molecules. Anything that's charged, so here we have ions, will not be able to make it across the membrane. If you're non-charged, you can go directly across. If you're too large, you cannot go across the membrane. Water is a special substance. So water is a polar molecule, but water also is able to go directly across the membrane. It doesn't do it very often, but it can move across the membrane. And when it does, it moves typically through protein channels called aquaporins. But when it can't move through one because there's no aquaporins, water will go directly through the plasma membrane. The cell size is going to vary dependent on the organism. Bacteria are always single-celled organisms, and they're very small. The multicellular organisms that are very large tend to have more, well, we have a lot more cells, and each cell is going to function with the other cells to allow us to do whatever jobs we have. Whereas bacteria, single cell, so within that cell, they're going to do everything. So here's multicellular organisms. Here we're going down to single-celled organisms, but these single-celled organisms, a human egg or a frog egg, is very small, but it's still something that could potentially be seen by the human eye without any help. If you do see this, it's impressive. You have really good eyesight. Typically, we use microscopes to see things that are smaller than a millimeter in size. And so this is where bacteria fall in. We can even see organelles that are found inside of cells, but we can't see the individual molecules that make up the organelles or the bacteria. And we also can't see viruses. Viruses are super small. So here, anything that's smaller than 100 nanometers in size, we're going to need an electron microscope to see. In this school, we don't have electron microscopes. We do have light microscopes, and they can allow us to see things up to 1,000 times bigger than what they normally would be. We will never see viruses with these microscopes, though. So I went online and I looked for something that I could use to show you the difference in size, because seeing that chart, that's good. It gives you some information. But I found this animation on Cells Alive, and basically you're looking at the head of a pin. So a little tiny pin has a head that's about 2 millimeters in diameter. And what we're going to do is we're going to continuously move in to closer to the head, magnify it to see different structures. So this is Cells Alive. Wonderful place. So we're going to look at, there's the pen, and right now we don't see anything, okay? We're going to look at all these different structures. Right now we're here. I'm going to just move up to 10. So at this point you can see the hair. There's something else there, but we can't make it out. We can see that beautiful hair there. So let's get a little bit closer. Oops, sorry. So here we can see the human hair. Do you see that? It's labeled. And we can see the dust mites. So those are dust mites. If you ever wondered if there was such a thing, dust mites are a thing. They're little microscopic arthropods. They're like little arachnids. And they are found in dust. They don't produce dust. We produce dust. The majority of the dust that we have in our house is actually skin cells that slough off of our body. But they utilize dust. And so if we have a lot of dust, there's going to be a lot of mites. And they can cause all kinds of problems. So now let's get a little bit smaller. So now we see something right there. What is that? Let's get a little closer. Well, let's see, what does this do? So now we're at 20 micrometers. Micrometer is 1,000 times smaller than a millimeter. And if we want to see where ragweed is this huge molecule, this huge structure. This is what causes a lot of our symptoms if we are allergic to pollen that gets into our nose and causes all kinds of problems. So, that's a lymphocyte. So a lymphocyte is a white blood cell that attacks specific pathogenic invaders. This is a eukaryotic cell. That's plant. Those are red blood cells. So red blood cells are beautiful biconcave structures, and they bring oxygen to our cells. So they're kind of big right there, but you've never probably seen an individual red blood cell like that. Baker's yeast. So this is a eukaryotic structure. Again, super small, but it is a eukaryotic structure. So here we've had eukaryotic structures all around. Plant, human or animal, I can say, and fungi. So let's get a little smaller. Let's get a little closer. Let's see, where's the E. coli? These are E. coli right here. So E. coli are bacteria that live in our gut. They're found in all animals. And some E. coli can cause a lot of problems. But E. coli are super small compared to our cells. There's Staphylococcus. So there are 4 cells of Staphylococcus all together. But each of them is an individual cell that can produce a colony of themselves. So each of these is an individual organism, just like the E. coli are individual organisms. So these are smaller than these. Smaller. Here, this is the Ebola virus. So right here we have E. coli, huge, and there's our blood. And this tiny little structure is the Ebola virus that causes hemorrhagic fever. So it causes us to bleed out, get a fever, our entire body just basically breaks down. Super tiny. Its shape is very similar to a special river, I believe. So very interesting. Then, did you see those little tiny things? Let's get a little closer. Boom. Those are rhinoviruses. So if we get closer, you can see them even better there. There's a bunch of them, but we get real close, you'll see that's a rhinovirus. The rhinovirus is what causes colds. Isn't that kind of cool? So you can see the size difference. We're looking here at the pin and here at rhinoviruses. So different cells are very different in size. Now remember, Ebola and the rhinovirus, these are not cell structures. They are non-cellular. They don't have a cell. So talking about cells and the size of cells, we have to have a certain, so all cells have to be able to gain nutrients and get rid of wastes. And the surface size of the cell is going to determine how effective it is at getting rid of waste and taking in nutrients. Larger cells, they need more nutrients and they're going to have to get rid of more wastes. So what happens is if the cell gets too large, it's not going to be very functional. Sorry. So if the cell is too large, it becomes non-functional, or you can't survive. And so we've evolved to become multicellular, and each individual cell can do more work than that one large cell. As volume increases, surface area decreases. And so we want to make sure that we don't have too large of a cell to get the job done. So let's look at a basic cell. This is a cube, and so let's just assume this is a single cell. It has the exact same surface area on all sides. To figure out surface area, you take side A times side B. So basically length times width, which is A squared, okay? So the surface area of a cube, you would take side A times side B, so A squared like this, but then you're going to add up the number of squares you have. So 1, 2, 3, 4, 5, 6. So it'd be 6 times A squared. The volume, you take length times width times height. And so that's going to be A cubed. And I do have here, let's assume we have a 1 millimeter square. So the volume would be 1 times 1 times 1, which would give us a volume of 1 cubed, or 1. Surface area is 1 squared, which is 1 again, times 6 sides. So that would be a 6. So the surface area to volume ratio is 6 to 1. If we change the cell size to 2 millimeters, the volume becomes 8, but the surface area only becomes 24. So 6, so it's 2 times 2 times 2, or 2 cubed, 8. 2 times 2 is 4, times 6 is 24. So when we do the surface area to volume ratio, that's 24 to 8, or a 3 to 1 ratio. So we've cut the surface area down by 1/2, or not by 1/2. We've cut it down in half. Let's do a 5 millimeter cube. So with 5 millimeter cube, we have 5 times 5 times 5 for the volume. 5 cubed is 125. The surface area is 6 times 5 squared, so 6 times 25, which is 150. And so that's an even smaller surface area to volume ratio. I'm going to stop here, and we're going to continue looking at different cell types and their basic structures, okay? So I'll see you in the next video. Bye.